Hello, everyone watching this. This is second episode of Antibuddies Journal Club. The first one was good. I love that paper. Apparently, some people didn't like that paper because it was too much metabolism. In this paper, we have got Tanu, who will be explaining about oncolytic viruses and their anti-tumor effects on DIPG and pHGG tumor mouse models. Anyway, I am Jatin and I am a PhD student. Tanu?
Hi, I'm Tanu. I am a PhD student in virology.
I'm Chaitanya Soni. Oh, it's okay. You go. You can go.
I'm Samriti. I'm also a PhD student in the field of oncolytic viruses and cancer.
Chaitanya?
He got stuck.
Oh, he got stuck.
I'm Chaitanya Soni. I just finished my medical school. I'm a physician. I'm practicing in India right now.
Nice. And
hello everyone. I'm Chaimo Sunri, and I'm just undergrad. I'm third year in biochemistry.
Nice. Yeah, Reach is the only undergraduate student in this journal club, so that means we get to bully him. Tanu, can you start? Let's go. I am slightly excited about this paper. I'm also hoping you will clear a lot of my doubts.
Okay, I'm sharing the screen now.
Mm-hmm.
So the second screen is not as an extension for you.
Have you started the presentation, Tanu? Is the PowerPoint open?
Yeah.
Yeah, I mean it should be coming on the second screen, but it is not.
Oh, if you open PowerPoint on the slideshow tab, there is an option where you can select which monitor you want to use. So while Tanu is setting up, Chaitanya, did you tell me once that you are interested in becoming a neurosurgeon? Was that it?
Yeah. I'm really interested with the brain, how it functions, and how it is still not been understood. We have so much technology available right now, but those technology has still not reached the research of brain.
Okay.
So it would be interesting to see the combination of the technology which is available right now and the brain functioning.
Okay.
So I'm very curious with that related to the research point of view, but clinically, I would love to pursue neurosurgery. Yeah.
Okay.
Okay, Jatin, I need to.
Okay, wait. Hold on a second. I'll help Tanu a bit. I'm going to disable my video. I'll be back in 1 minute. All right, I'm back. Let's go.
Okay. So the paper we are presenting today is Oncolytic virus Delta-24-RGD elicits an anti-tumor effect in pediatric glioma and DIPG mouse models. This was published last year in Nature, and let's see what it says. So a little background about the two types of pediatric brain tumors we'll be talking today. First one is pHGG and the second one is DIPG. And both of them are just in 8 to 12% of all other pediatric CNS tumors. So there can be two types of tumor, central nervous system and peripheral nervous system. This one occurs in the brain and spinal cord, and it occurs in children in the range of the age 6 to 9. It is called an orphan disease because the occurrence of this disease is very, very rare, and hence not many studies has been done on that. It occurs in around 85 out of 100,000 children. There are two types of treatment which are mentioned in this paper for the two types of tumor. For pHGG, so pHGG is pediatric high-grade glioma and DIPG is diffuse intrinsic pontine glioma. And for pHGG, the treatment is a surgery where you remove the tumor, and it is followed by radiotherapy or temozolomide cycles. The radiotherapy and the drug are supposed to reduce or get rid of any other remaining tumor cells which are not removed during the surgery, but the surgery is supposed to remove the main cancer stem cell part of the tumor, so the tumor cannot spread anymore then. And pHGG by this technique can ideally be like curative. But on the other hand, the DIPG, there is no treatment for that. There is only radiotherapy, which is not curative because radiotherapy can only reduce your tumor, but not reduce the cancer stem cells, which are shown in red here. And the cancer stem cells can then give rise to many more cells in like a few days or a few months, depending on the type of tumor. So there is no cure for DIPG yet.
Tanu, I have
When
a
you
question. Can you
Yeah.
go back? So do you know, is there any morphologic difference between the two tumors? Do they look different? How are they classified differently? I'm not 100% clear.
So this was the slide where I was.
Oh, you were going for that? Okay. And one more
Yeah,
thing, one more thing while we're at the back. Are you going to talk about cancer stem cells in future? In that case, I'll just let you.
Not in this presentation, because it is not much about cancer stem cells here. It's more about the oncolytic part.
Can you briefly tell us what are these cancer stem cells? Why are they important?
Yeah. So every cancer will have two kinds of cells. One will be the cancer stem cells, which are generally towards the very center of the cancer or tumor. And secondly, there will be other cells which are grown out of the cancer stem cells, which are surrounding and towards the periphery of the tumor. So this cancer stem cells or tumor stem cells, sorry, cancer is when it is metastasized. So the tumor stem cells can give rise to many more kinds of tumor cells. Even like one tumor can give rise to another kind of tumor cells, but they are very hard to get rid of. So chemotherapy or the radiotherapy or multiple drugs do not act on these cancer stem cells. They can kill the other cells which are on the periphery. And when these cancer stems are metastasized and move to other parts of the body, they can attach to the other part of the body and give rise to many more tumor cells, and that's how it becomes a cancer throughout the body. Okay, I'll continue then.
So can I ask a question?
Yeah.
I would like to know how do these cancer cells normally metastasize? Like what is the pathway they use? Is it by blood or by lymphatic?
I know solid tumors, they do by lymph nodes mostly. Maybe any tumor of the blood can also metastasize via the blood. But solid tumors generally use the lymph nodes.
Okay.
But I thought even solid tumors can go through epithelial to mesenchymal transitions and they can get inside the blood, right?
That's when they are doing the transition.
Okay.
Yep. It's like cancer. Any stem cell actually can turn from one to the other type of differentiated cells. Not going too much away from the point, I'll come back to the difference between the two pediatric brain tumors. The first one is pHGG. This is a high-grade tumor, which is a high-grade glioma, which is spread throughout the brain. So when you take a section of the brain like this, horizontal section, you can see the tumor spreading all over the periphery as well as towards the center. And this is like a grade 1 pHGG, and this is a stage 1 pHGG, and this is a stage 3 pHGG. Whereas in the case of DIPG, this is much more concentrated and it is only present in the brain stem, which is this part of the brain shown in 3 different planes.
Then what are these images? Are these?
These are MRI.
Okay.
Does this answer your question? The differences? Okay. So this is what we can see morphologically, but functionally they are both termed as pediatric brain tumors, which are almost similar.
One more question. So glioma, these glial cells, are they every cell except the neurons inside the brain?
I don't think so. Like, I don't know much about brain morphology, but I think glial cells is a different. Yeah.
I think glial cells are the defense cells. There are different kind of cells like astrocytes, glial cells. These are one category of cells. They help in support of the blood-brain barrier. So if there, like, brain is very protected from the blood and other particles in the body. So our body can't easily contact the brain. These glial cells, astrocytes, they help in protecting the brain, making a barrier. That's where glial cells come
Okay.
come in.
So glial cells isn't this a generic term? I might be confusing here, but glial cells isn't it a generic term for astrocytes, microglia, and one more cell that I'm forgetting in the brain? Everything except the neurons, or are these specialized cells? Like, is this an umbrella term, or is this a specialized cell type?
These are
That
specialized cell type.
okay. Okay, then I
Like
I,
if
I
you have heard of macrophage, in our immune system we have macrophage, but macrophage are not same in our brain. They specialize and they have a different name. That is microglial, I think.
okay. Anyway, so this glioma would be any tumor of those glial cells.
Yeah.
Okay.
Okay. So there are a few viruses which love tumors, and why they love tumors is they love replicating inside the tumors. Tumor cells, they replicate continuously. So it's like a continuous host which will not die, it will stay in place forever. And the second reason is that tumor cells usually have mechanisms to evade the immune system of the host. And this is advantageous for the virus because the virus wants a microenvironment to grow where there is no worries of the immune system coming and attacking it. So tumor cells are the best for it. Oncolytic virus can infect and kill tumor cells, and this occurs in three stages. The first one, where the viruses preferentially insert and replicate in tumor cells, which is right here. So the virus needs to attach, enter the cells, and as well as replicate in those cells. Secondly, the second step is that where viruses lyse the cells to release its progeny. And these progeny viruses now can infect nearby tumor cells or travel to other parts of the body to infect the metastatic cancer cells. Thirdly, they can third part is that where the lyse cells present in tumor antigens for in the microenvironment for the host immune system to come, infiltrate, and attack the whole tumor. So when the cell is lysed by the virus, every tumor antigen inside the cell is now present in the microenvironment, and the host can give the anti-tumor response um, likewise. And that's when the host immune system takes over and the work of the virus is done. And after a period of time, we don't want the virus anymore in our body. So the oncolytic virus must also have a mechanism through which it removes itself from the body. That's how, so this is a mechanism for which oncolytic viruses are also used in therapy these days. But there are a few modifications done in the oncolytic virus, which I will refer to in the following slides. A little background about adenovirus. So if the virus can attach to the receptor and enter the cells are called to be susceptible to the virus. Now, if the cell has necessary elements for the virus to replicate and form viral progenies, then the cell is called permissive to that virus. For a successful viral cycle to take place in any host cells, the host cell must be both susceptible as well as permissible to the virus for attachment, entry, replication, translation, and forming of the assembly of the viral particles in the end. Um, so this paper talks about a protein called E1A. For that, I'll give you a little background about the different types of mRNA and the proteins released by adenovirus during their infection. So adenovirus doesn't release all the mRNA at the very same time. They have a temporal release of their genes. In the very first, they release the immediate early genes, which form immediate early RNA, then early genes, and then late genes. Immediate early genes prepare the cell for DNA synthesis by activating the cellular genes that regulate the cell cycle, enzymes for DNA precursor synthesis, and proteins for the initiation of cellular DNA synthesis. After production of this RNA comes the production of the immediate early proteins, one of which is the E1A protein talked about in this paper. And this E1A stops or regulates multiple things, mainly the RB pathway in the host, which helps replication, which helps start the replication of adenovirus in those cells. Secondly, early genes assist in viral transcription and replication. And finally, the late genes are generally structural proteins, genes producing structural proteins, which are used in viral assembly. So this will be like the protein in the present in the capsids, or glycoproteins which are incorporated in the envelope of the virus, or any fiber proteins or any such structural proteins are produced by the late RNA.
Tanu, I have a question.
Yes.
This is a double-stranded DNA virus, right?
Yes, it is.
And is it doing all of this inside the nucleus?
It does this parts inside the nucleus, yes.
So the E1A.
And then the
Mhm.
so every time it produces the protein, the RNA needs to go out of the nucleus, produce the protein, and then there is a machinery by which the proteins come inside the nucleus again, and helps in translation or replication. And
Okay.
finally, only in this stage, it is like completely out of the nucleus.
This E1A and E2 are viral proteins, right? And they are helping the virus carry this process out in different stages.
Yes.
Okay. And so you might not know the answer to this, but do you know why it would do that? Why there is a need for multi-stage transcription?
It is not in all the viruses. Some viruses have much simpler mechanism of action, but some viruses such as herpes simplex virus or adenovirus, they generally release their genes in a temporal manner. And I don't know don't know the exact answer for the temporal release, but I would guess that they don't want like multiple proteins altogether in the cell or the nucleus, so that not all the functions are taking place at the same time, but one function is taking place at a particular time, the function is ending and then helping start the other function.
Okay.
That's my guess. Okay. Then we come to the replication competent adenovirus, and the virus which is targeted here is Delta-24-RGD, which I'll be explaining more about. A replication competent virus can have two changes, which the oncolytic virus parent doesn't have. The first change, these changes are artificial, done in the lab. First change will be the viral virulent genes responsible for viral replication in normal cells are deleted. So when we are saying these are oncolytic viruses, it doesn't mean that it cannot at all replicate in normal cells. They can replicate in normal cells as well, because the viruses itself contains some genes which help mutate or which help stop the normal functioning of normal cells and help in replication of the virus. But in cancer cells, this kind of genes are already expressed. So the function of the oncovirus genes now becomes redundant. So you don't want two proteins doing the same function, which are basically the same. So now you can delete those genes from your oncolytic virus so that they cannot go and disrupt normal functioning of normal cells, but they can easily replicate in the cancer cells because the cancer cells already have the genes which are making them which are helping the virus replicate.
So
one of these genes, yeah.
Okay, no, continue.
Okay, the second change. Okay, but basically these viruses would replicate in cells where the RB pathway or P53 pathway is mutated. And those are already mutated in cancer cells, as I told you. Secondly, these viruses changes are made so that this virus target tumor cells specifically, and such that, I mean, in the example I'll show you for this virus itself, what change can specifically target them towards the tumor.
Okay, yeah. Now I'll ask. Tanu, you said this
Yeah.
virus has a part, the 24 base pair is deleted in E1A that's required. That E1A it guides the first phase of transcription, right?
E1A prepares the cell for replication of the virus.
And it's also binds to this RB retinoblastoma gene.
I didn't come to it yet.
Oh, you didn't? Okay. I thought you might I might have missed it and you said it. Okay.
No,
So
I'll say it. So the name is very self-explanatory, but the 24 here refers to 24 base pairs, which are deleted from the E1A gene of the virus. And these 24 base pairs were responsible for replication in normal cells, but now when it is deleted, it is more selective to replication in tumor cells only. E1A gene. So first, let me tell about the RB pathway. The RB is attached to E2F and DP when cyclin and CDK, which are host proteins, come in function. The RB is phosphorylated and it dissociates from the complex. Now, E2F and DP are free to go in the nucleus and do transcription of the genes, which lead the change of cell from G1 phase to S phase. So in the G1 phase of cell cycle, the cell is only growing and preparing for replication replication. But in the S phase, the cell replication and forming of multiple cells from the same cell is happening. This is basically done by cyclin and CDK in the host. But when the virus is there, the virus wants continuous cell replication for their own need. So in that case, the virus itself produces immediate early gene, which is E1A. And this cell, this protein can bind to RB, has more affinity than this two to bind to RB. And that's how they make this E2F and DP free for transcription of S phase genes. Yes, Jatin, any other questions?
so it's doing the same thing as phosphorylation, right? As in similar to phosphorylation, it's preventing RB from binding to ETF and DP.
The main consequence is similar, but in this case, it is just like it's a dissociative binding. But in the phosphorylation of RB, it is just like separated.
Okay, so I know you said this just now. I'm still not clear. How is this 24 base pair deletion affecting its affinity and its effect in tumor and non-tumor cells? I'm not clear about that.
Okay, so this diagram that I have here is for non-tumor cells. In non-tumor cell, the E1A, in absence of S phase starting factors such as cyclin and CDK, can go and start the S phase for the cells. Now, if the S phase is started, many other cells will be produced, and the virus can keep on replicating in different cells and exponentially producing their progenies in those cells, right? So this is for a normal cell. But in a tumor cell, this cyclin and CDK function is already upregulated. So you don't want E1A functioning in that tumor cell too. So the important genes which are responsible for binding to RB is now deleted. So this is basically non-functional E1A in the oncovirus right now.
Oh, I see. So does this mean that this E1A is required? It's like E1A is pushing the cell into cell division, replication, and without E1A, the virus will only replicate if the I mean it's viral replication will only work if the cell is already going through rapid cell cycles.
Replicating, yes.
Okay,
Yes,
so
correct.
that means slowly dividing cells will be less affected, but they will still be affected, but just not as much by this virus.
Yes, and we would expect that it is like almost not affected. Like there has been studies showing that they are
In an
almost
ideal
not
world.
affected. But for this Delta-24-RGD, it has been shown that it is non-cytotoxic to normal cells.
And what's the RGD about?
Yeah. So the RGD is a small amino acid sequence which helps the virus interact with two types of integrins, alpha gamma beta 3 and alpha gamma beta 5. So if you can see, there are three chains forming an integrin. The alpha gamma and the beta separately are two subunits, and the amino acid RGD attaches very well to this integrin in this diagram, and that's what they took for their study. So if they include the amino acid sequence RGD in the oncovirus that they are using, which is adenovirus, this will specifically attach to the integrins which are already overexpressed in gliomal cell tumors. So this is another factor which is adding to the specificity of oncovirus to gliomal cell tumors.
They have added this sequence specifically to virus?
Yes.
Okay.
Yes.
I have a technical question. Where do you see the presentation?
Hmm?
Uh,
You cannot
Chetan,
see the presentation?
Could you hear me?
Chetan, you're cutting out
Yeah,
for a
you're
second.
cutting. Sorry.
Yeah, I can't see the presentation.
Oh, are you in your phone? Oh,
It's okay, never mind.
okay.
Okay. Okay. Coming to the advantages of using this oncovirus is it's been already shown in clinical trials for tumor studies that it is self to administer intratumorally. And Jatin, that also answers your question that why in normal cells it is not affecting at all, because we are injecting it intratumorally, and
Mm-hmm.
after its function is done, it is not found anymore in any other cell or fluid of the body.
Yeah, I'll just point out one thing that they mentioned about this virus, what it does. We also discussed about that in a podcast, the last podcast we did on nano immunotherapeutics. There is this thing called immunogenic cell death, and that's a big proportion of how these viruses are helping. Not only are these viruses killing the tumor cell, they're also making their fragments or debris available to the antigen presenting cells in the nearby lymph nodes that can take it up and they can present it to T cells. So overall, these viruses, among a lot of things that they're doing, they are helping the immune system itself in preparing for fighting these tumors or recognizing these tumors.
Yes.
Do check out the last episode; it's really good. I'll just plug it in.
Yeah. I also mentioned that in the background of this paper that in the very last stage, they lyse the cells and release all the tumor specific antigens in its microenvironment for the immune cells to recognize. Okay. And secondly, this is one of the oncoviruses which helps in assisted immunotherapy by itself. Generally, what we do is a combinatory therapy where a oncovirus is combined with some cytokines or some stimulatory factors for immune cells, and they are used together for the therapy of a tumor. But this virus by itself can increase number of uh, cytokines in the tumor microenvironment, and it itself helps in assisted immunotherapy. Okay. So starting with their experiments, the first experiment they did was an in silico experiment to check if the receptors for Delta-24-RGD were present in the patient samples. They collected 220 patient samples, and basically there are 4 receptors by which this oncovirus can attach and enter into the cells. These receptors are integrin 3, integrin 5, integrin alpha gamma beta 3, integrin alpha gamma beta 5, integrin alpha gamma beta Coxsackie adenovirus receptor. So 4 receptors. This first study is to see if the cells are susceptible to the virus, and they see that for almost all, not almost, for all the molecular genomic group, so for all the 220 patient samples, the receptors were expressed in those tumors either in low abundance or in high abundance, but basically they all were the receptors were present in all the tumor cells.
Can you go back a second?
You are muted, Jatin.
Oh, yeah. Can you hear me now?
Yes.
Those 4 types. So are they those 4 colors? They are subtypes of this glioma, right?
They are, yeah, difference in molecular function, I
So
think.
that green and dark green, they look like some epigenetic markers.
Yeah, they do. They look like methylations.
At Samriti,
This was how.
which tumor do you work on?
Sorry.
I work on sarcomas. So
Wait, are those epithelial muscle cells?
those are mesenchymal muscle cells.
Oh.
Well, I work on rhabdomyosarcoma, which are the muscle cells, but there's a variety of different subtypes that fall within sarcoma.
Okay. I was just wondering how do they grade these, but must be very complicated. Tanu, you wouldn't know how they are differentiated,
sure.
right? I wouldn't. I wouldn't blame you for not knowing. How are these 4 different?
What I guessed is that out of all the patient samples they collected, they created 4 subgroups based on one common features they had. So I think when they say 3.3 K27M, all the patients in this subgroup will have this very methylation pattern. That is what I guessed.
Okay.
Maybe different. Okay. So as we previously discussed, cyclin D and CDK4 and 6 phosphorylates the RB, which helps in progression of cell cycle to S phase. And along with that, we have another host gene, CDKN2A, which produces 2 tumor suppressor genes, P14 and P16, which either slows the cell cycle or results in apoptosis. And in the next experiment, they use all this molecular factors like CDK4, 6, cyclin D, and the gene CDKN2A.
So, Tanu, that is a cyclin dependent kinase inhibitor 2A, and it gives rise to both of those proteins, P14 and P16.
Yes.
Yeah, I just looked at it. Okay.
Yeah, thanks for expanding it. Now, for the cells to be permissive to the virus needs to be able to replicate and form viral progenes inside the cell. The Delta-24-RGD virus requires the RB pathway for replication, as we already saw. And so the first thing they saw was if the genes involved in the RB pathway had any mutations like amplification or deletion in these tumor cells. As I told, the virus now don't have the capacity to infect any cell where RB pathway is functional. So they need to be sure that in this tumor cells, the RB pathway is not functional. They saw that both CDK and CD4 had modest level, which I don't clearly understand why they said modest level, because here the amplification is not, like, there is no amplification at all in some of the subgroups of patients. But I'll go with whatever they are saying, that CDK and CD4 had modest level in all molecular groups. The cyclin D1 amplification was only seen in one of the subgroups. So basically what they're trying to say is there was not much difference in the cyclins or CDKs. There was one RB deletion in IDH1 subgroup of patients, but the interesting thing that they observed, was that this gene CDKN2A was deleted in almost all the subgroups of patients. And that's how they gained confidence that yes, in these tumors, the RB pathway is not functional because of disruption of CDK and 2A, which will lead to disruption of two tumor suppressor genes, P14 and P16. In their next experiment, they are doing some in vitro studies in different tumor cell lines. And all of these tumor cell lines are either from pHGG tumors or DIPG tumors. Firstly, the abundance of integrins and the Coxsackie virus, adenovirus receptor, which is also known as CAR, here was tested in different cell lines, and the CAR was seen to be highly expressed in all the tested cells. So now, not only in silico, by in vitro analysis too, they confirmed that yes, the virus can attach and enter because they have abundance of this receptor present.
Do they compare it to any normal cells?
That's what, like, I thought that they didn't keep any normal cell control here. Yeah, that's strange. But they say that they are highly expressed in this kind of tumors. So I would assume they did some preliminary study where they compared it to the normal cells, but they don't show it in this paper though.
Because I think adenovirus normally infects like epithelial cells in the lungs, right? So
Yes.
that would be a good comparison to show. I don't know how that translates to a mouse model, but yeah.
Yeah. They also mentioned this in the very end, that adenovirus are not known to infect mouse cell lines very well.
Yeah.
But somehow this virus did, luckily, so they can carry on their studies. Yeah. So secondly, they tested different MOIs. Here is MOI 10 and here is MOI 100 for different cell lines. MOI is multiplicity of infection. That is how many viruses you're giving per cell. A replication deficient adenovirus was used, which is this one, which had GFP labeled, and they were protection in these selected cell lines. The 10 MOI led to above 50% of infected cells, whereas the 100 MOI led to it's like perfectly 100% cells infection being carrying on. Another thing observed was that the pHGG cells were more permissible than the DIPG cells in general in vitro. Next, they wanted to see if the replication is taking place well, and they're giving rise to proteins. So they tested two proteins, one immediate early protein, E1A, and secondly, a late protein, fiber, which is found in the structure of adenovirus. So E1A was very well seen in Western blots. So the replication is taking the proteins leading to replication are being expressed. And secondly, the fiber protein was also seen in Western blots, which confirms that the replication took place, transcription took place, and finally the virus is can go and be assembled into a virus particle.
Tanu, you said fiber is a structural protein, right?
Yeah,
So
so it
it's
is a late
late
phase
phase.
protein.
Okay. And GRB2, what is that again?
It is a control. It is growth factor receptor bound protein 2, just like actin controls.
Okay. Except that I don't like actin as a control, especially for tumor cells. Actin would be the most horrible control. And even GAPDH is a horrible control. I recently read a paper that GAPDH does a lot more than just being a part of the glycolytic cycle. It binds to interferon gamma mRNA. Very weird, right? So if you have a lot of inflammation going on, you could have increased or decreased GAPDH just because of that.
So it's not a good control
No,
that way.
not for immune like immunological studies. No, and actin. I feel like actin is not a good control for anything. It's not for cancers, not for immune cells, because immune cells also go through rapid divisions, and that would change your actin amount in
Yeah.
the cell.
I have used tubulin with neuronal cancer cells as a control, but
Did that work well?
that used to work well, yes, but I generally praise the antibody for working well in a Western blot, but I
Okay.
didn't compare it with any other controls, so I used only tubulin in those studies. Okay.
Do you
So
know
here-
why they chose these 4 cell lines to study a little bit further after looking at that receptor expression panel? Or did they do all of those cell lines in maybe the supplemental?
There are some supplementary figures which have all the cell line present, but mainly they are using some representative of pHGG and some representative of DIPG out of all the cell lines. And I'm not clear the reason why, but
because like if you go back, I think TP54
they have, if I go back, there are multiple
doesn't
cell lines.
yeah, like TP54 is like mediocre, I would say, right?
In which case are you talking about?
In the expression of the alpha V beta 5.
Okay. Yeah.
Yeah, so they chose the SU, which is high. They chose PBT, which is high.
Hold on, 54 is good. Hold on, 54 is good.
54 is good, yeah.
54 is good.
So they chose the top 3.
Yeah, maybe that.
Yeah, but in also some studies, they are using the CHLA03AA, which is lower. So they are just using a few representatives, which I think worked very well for them.
The naming is so random. I hate when cell lines are named like alphabet soups. Random things that come into people's mind, just put them in.
Okay. Then they see that there was at least 2 log increase in virus from input virus, which is 10 to the power 6 PFU per ml. They saw at least there was like 10 to the power 8 output amount of PFU per ml virus. And again, it was noticed that in general, the pHGG cell lines were more permissive, had better viral outputs than DIPG cell lines.
Tanu, just for the sake of explanation, can you tell everybody what is PFU per ml? What are we counting here exactly?
Yeah. So PFU per ml is plaque forming unit per ml. One plaque would represent one infectious virus. And when you are growing virus, there may be multiple viruses which are not infectious because viruses continuously grow through mutations, especially the RNA viruses. And this is a double-stranded DNA, so not confusing with this one. But PFU per ml will be out of all those viruses, the number of infectious viruses per ml. basically one infectious virus can lead to one plague, which is basically multiple dead cells or disrupted cells in a cell culture dish.
The calculation is similar to CFU per ml for bacteria, right? It's instead of counting colonies, you're counting dead masses of cells. Okay.
Yeah, you're counting the plagues instead of that. Here they conclude that Delta-24-RGD is able to infect and undergo a viable viral cell cycle. So viral cycle, sorry, in pHGG as well as DIPG cell lines. In the next study, so now they know the cells are susceptible as well as permissible to the virus. Next, they wanted to see if the virus is causing any cytotoxic effect in those cells or not. For that, they do a IC50 study. IC50 is inhibitory concentration where 50% of cells are dead. And it was in between 3.6 to 68, which is a very wide range of MOIs in DIPG cells, and 0.8 to 32 MOI in pHGG cells, which is also a huge range of MOIs. So Samriti, I don't know if you work on something similar, but do you also face like such a huge range of MOI as IC50s?
Honestly, I'm not too sure. Because they're working on a lot of different cell lines. I'm not too sure.
Yeah.
Yeah.
Because I don't work with so many cell line all at once. But yeah, exactly. But when we work with one particular cell line, we only get like one value of MOI, which is IC50, right?
Yeah.
So that's a bit strange. But yeah, anyway, they found ranges of IC50 values. And secondly, in this figure, they show that the cell viability was accessed during using MTS assay after 5 days of infection, and the cell proliferation percentage was measured for different MOIs. And after 5 days, there was a very much reduction in the number of tumor cells in all 4 cell lines that they tested on.
Tanu, I know it's too late for this, but did you explain what an MOI is, just in case anybody doesn't know?
Yes.
Okay.
But again, it's multiplicity of infection, which is how many viruses per cell you are giving.
Going back to the whole variability in the IC50, if they're seeing that much variability just in vitro in the cell lines, I'm just thinking like how much variability you're going to get in patients, right?
Yeah.
Because there's already so many subtypes that they tested in the beginning.
That was my concern too, that the range is like so wide.
Yeah.
No, yeah. Maybe they need to do like more personalized studies, seeing which type of tumor cell lines the particular patient will have and decide on the MOIs like that. But it's difficult, but not impossible.
Yeah, that's very difficult to do. Yeah.
Yeah.
Finally, good stuff.
In this, so after all these studies, first I wanted to check if the Delta-24-RGD are working on early tumor mouse models. So in early tumor of mouse, they inject tumor cells inside the mouse, and 3 days after injecting the tumor cells, when the tumor is in very, very early stage, they infect 10 to the power 8 PFU per animal of the oncovirus, and that's when the treatment starts from the 3rd day. And then mouse were
Tanu, I have a technical question. I'm sorry. So you can only measure PFU per ml for cells that adhere, right?
Once you know the PFU per ml of the virus, you can calculate how much PFU or infectious viral particles you are going to give the animal.
No, I mean, if a cell, so you perform the PFU per ml experiment on an adherent cell line, and then you look for
Yes.
holes. What if you have suspension cell line which don't adhere? Can you measure? How would you measure PFU per ml for that?
I have never done that personally, but I would assume that because they are keeping the cells at the very lowest level or just above the plate, and then they are doing a agar overlay, the suspension cells will also remain somewhat adherent
Oh,
to the plate.
do they immobilize them with an agar?
For the adherent, yeah, they always
Oh,
use the
okay, okay.
agar overlay. Yeah, that.
Okay. I didn't know that.
Yeah, but I've never heard of like I say on suspension cell lines, so I cannot give a exact protocol for that.
Because if you try it on a blood tumor, leukemia, lymphoma, those cells are not going to be adhering.
But I think the agar overlay somehow helps them immobilize
Yeah,
the
I have
surface
never
of the
done this,
plate.
but if in my imagination it looks like that agar will immobilize them.
Yeah.
Well, usually when you're looking at how many infectious viral particles you have, you're trying to use a cell line that is very, very susceptible. So that would be your most positive control, essentially,
Oh.
right? So
Yes.
that will tell you with more accuracy how many infectious particles you actually have, and then when you inject them in a mouse, you're obviously going to get less infection than you would in vitro. So it's just the in vitro is giving you a ballpark most likely number of infectious virus that you have.
Okay.
Yeah,
that makes sense.
And also for adenovirus, they need higher PFU than other viruses such as norovirus. That's why they used 10 to the power 8 PFU per animal for this, but generally for other viruses such as norovirus, which is a RNA virus, we can just use like 10 to the power 5 PFU per animal.
Is that generic that RNA viruses are more infectious than DNA viruses?
Yeah, I have not studied about all of them, but
Okay.
yeah, I cannot say generically. Sorry. Okay. The mouse were monitored till the weight loss or the loss of control of bodily movements were observed. And in all the cases, the treatment with the oncovirus showed better survival of the mouse. Out of the 4 cell lines they are showing here, in the 3 cell lines, they can see long-term survivability of the mouse too. In just 1 cell line, sorry, not for the mouse, long-term survivability of the cells, but just in
You
one
mean
case.
animals, right? These are animals that have been given
Oh yeah, sorry.
orthotropic transfer of these cell lines so that these animals will develop a tumor.
Yep.
Okay.
I got confused. So there was long-term survivability of the mouse in case of when 3 of the cell lines were injected. Only in case of 1 cell line injection, all the mouse died, but still the survivability was better as compared to control.
Yeah, that TP54 looks very shady. If I have that tumor, I'm not getting this virus for my treatment.
Also, this is in nude mice, which are
No, these are not in nude mice. The nude mice experiments, I think, are coming up.
Okay.
No, this one is in nude mice. It's figure 3,
This
right?
one is?
Yeah.
Yeah.
Oh, okay, that makes sense. So just for
Because
anybody who's
these-
watching,
oh.
Can any one of you explain what is the difference here, and what are we looking at in a nude mice compared to a wild type regular mice?
Yeah, they're just they don't have a thymus, or they have like a dysfunctional thymus, so they don't have generally it's T cells, so they have an impaired immune system. So this allows for the study of human cell lines, because human cell lines you can't put in a mouse. So they use these nude mice to be able to grow those humanized cell lines in vivo and study those. But then you can't really do a lot of immunology studies because they don't have that functional adaptive immune system.
And this would also not be representative of what you would find in a healthy individual, because in healthy individual, so right now you're looking at the effect of these viruses without any immune response, as in all the immune response that would entail towards the tumor as a result of this viral infection. So you're just looking at what the virus does to the tumor
by itself.
by itself, or by anything except T cell responses.
Yeah. So this, I think, should be taken as with a grain of salt, honestly. Yeah. This is the result is not very surprising.
Mm-hmm.
Yeah, that makes sense. I didn't notice that it was on nude mice, but now it makes sense that the virus itself is doing whatever changes it is doing right here. The immune system response hasn't come in yet. So, yeah.
Yeah, you would expect a better response if you do it.
Yeah, much better.
Yeah, but let's see.
Let's see. And they also did some pathological analysis, and they said that I don't know much about pathological analysis, so if anyone else can jump in, but they said that for this kind of tumors, they showed some midline glioma in TP80 model, and tumor cells were shown to infiltrate the brain parenchyma and cerebellum.
I'll pretend that I know what exactly that is.
But what I can see from this figure is, I think these are the areas and these are the areas where they are assuming the tumor to be.
Hold on, isn't the darker color supposed to be cell growth? Like I'm assuming this is a dye that's staining the nucleus.
Mm-hmm. Yeah, darker will be more proliferation.
Cell nuclei, right? Okay.
Okay. But right here, you can see very well difference in the amount of tumor volume in control cells versus the cells which in control mice brain versus the mice which was treated with Delta-24-RGD. The tumor volume is reduced, but they did not show at a later time point if the tumor volume is completely destroyed or not, which I would have loved to see.
It is reducing. With my limited knowledge of pathology, I can see that it's reducing.
Yes. Yeah, visually we can see that. Next, they tried two different immunofluorescent stains. One is hexon. Hexon, again, is a late protein, which is a coat protein of adenovirus. And E2A, as we know, is a immediate early protein.
E1A.
Sorry, E1A. And their expression was found in virus-treated mice, demonstrating that the reduction in tumor is being carried out in presence of virus. So the virus protein expression was no longer shown by long-term survivor mouse, which they do not show here, but they say so I'll believe them, which is a good sign for oncolytic therapy because we don't want the virus to stay in the body forever.
Next, Tanu, can you explain H&E?
I forgot what H stands for, but it's
hematoxylin.
Hematoxylin and eosin stain. Jatin, I would want you to explain that.
Oh, okay. I'll do that. If what I remember, hematoxylin stains. It's a differential stain, so the charged molecules inside, like DNA, they will take the stain darker, while uncharged molecules outside, apart from DNA and some proteins, they will not take the charge stain as much. So you can again differentially look at nucleuses in this. The nucleus appear darker, the non-nuclear compartments appear lighter, what you're seeing here is that more dark spots means they're more very likely individual cells.
Mm-hmm. Strangely, they don't compare the hexon and E1A with the control, which we would expect that there is no staining in the control at all, but at least it's good to see that. Anyway, in the next figure, they check the survival of the mouse, and this is of the mouse which had established tumor. So in this case, the tumor cells were injected in the mouse, and after 15 days of implantation, the treatment was given. And in 15 days, they expected that the mouse will have an established tumor. All the studies prior to this was done on early stage tumor, which is just after 3 days. And they saw that there was 33% survival in case of mouse implanted with this cell line, and a better survival for mouse with this cell line.
So again, they have to be nude mice, right?
They have to be.
Okay.
By these experiments, they told that in vivo administration of the oncovirus Delta-24-RGD decreases tumor and increases the survivability in mouse. So just stop me if you have any questions, because I'm continuously talking. Okay. Now comes primary cell line experiments. XFM and NP53 are primary tumor cell lines from tumor arising in pons of genetically modified mice. And these cells are 100% susceptible to adenovirus infection in MOIs as low as 10.
So far they have shown it to somewhat work in human cells and somewhat work in human cells that were given to nude mice, right? That's what they have shown us in the past two figures.
They have shown that human receptors, the human cells have the receptors and everything. And I'm not sure whether those cell lines were human or mouse.
No, they were human cell lines, right? That's why they were using the nude mice. And they have, so far, they haven't shown any primary cells from patients, right?
They haven't.
Okay, and this is primary cells from mice.
Yes.
Okay, because you said cell lines, so I am getting confused. Is it's primary cell or?
These are they call them cell lines. So I would assume that there is a genetically modified mice from which you take primary cell lines, cells, but those cells also have a cell line name to them.
As in, are they immortalized? Possibly. Or they might be immortal cells by themselves. They're cancer cells. Okay.
Maybe.
Okay.
So here they see that in primary mouse tumors, MOI as low as 10 works for 100% susceptibility. And adenovirus replication is generally hindered in murine cell lines. But for these cell lines, expression of the fiber protein, which is a structural protein, was determined at higher MOIs, showing proper replication is taking place of the virus. In fact, at 300 MOI, virus PFU per ml went significantly higher after 3 days as compared to control, which is the input virus. So basically, the infection and replication of virus is taking place in primary mouse cell lines.
What's the control again? You said input virus?
Yeah, they say they represent the input virus as control here.
What's the input virus without the RGD and 24 deletion?
The input virus is the same virus, but it's how much amount you are injecting in those tumors. And this is the output virus, this 3 bars. I don't understand what exactly are you asking.
I mean, what does the control here mean? Oh, when you say input virus, as in, oh, do you also take the virus sample? Like, you do take the supernatant again to check how many viruses are there? Is that what do you do when you say input and output?
Yeah, so this assay is showing the titer of the virus after it's been injected in either this or this cell line. And to check the titer, you need to input some sample or supernatant or anything. And they say that they inserted this much amount, and this is what came out as output.
Okay. I am very bad at virology, so I'll
So
accept that answer.
I think what they have these cell lines. They plated these cell lines.
Mm-hmm.
They put in an input virus of whatever was it, 10 to the 6, I think.
It is 10 to the 5. 5
10
for one and 6 for one, I guess. No, 5 for both.
10 to the 5. So that's how much virus they know is there.
Yes.
They know 10 to the 5 virus particles will be in this on top of these cells, and then they allow it to infect for whatever period of time.
3 days here.
5 no, not 5 days.
3 days.
3 days, yeah.
Yes.
And then after 3 days, they take all of the supernatant off of the cells, and then now they're quantifying how much cells are still remain, how many viral particles are in the supernatant. So it will tell you if it's close to 10 to the 5, it would say that means the virus was not infecting and was not replicating, was not budding out.
Okay.
Yes.
So that's 300 means it's 300 times more than what was input before. Right?
Yeah, these are just the ratio of virus to cell.
Okay.
So
Okay.
if you put, so in MOI of 50, they're putting 50 virus particles per cell.
Okay. Okay.
So the greater MOIs that you have, the more likely it is for a virus to infect a cell, because 300 is basically a ratio of 300 virus to one cell. So it just increases the likelihood that that cell will become infected.
Okay.
So
and finally,
a 300 MOI is very, very high. Very high.
it is very high.
Yeah.
And that's why they, at the very start of this experiment, they say that mouse cell lines are not susceptible to this virus. That's why they're using such a high MOI. But still. So Jatin, finally they see that there is 1 log increase in viruses. So basically 100 more viruses were found, and this 100 new viruses were produced by replication in the cell lines. That's why they say that these cell lines are susceptible as well as permissive to the virus.
Is that tenfold or 100-fold? Isn't that
It's 10
tenfold?
to the power 5 to 6, right?
So that's tenfold, right?
Oh, it's 10. Yes. 10 more viruses.
Yeah, and scientists are supposed to be smart. Sometimes we forget numbers.
Sorry.
No, I'm kidding. I make stupid mistakes all the time.
Okay. In this figure, virion assembly was observed in the cell nucleus, which are the darker spots right here, and in magnified right here and here. These are icosahedral viruses, which will be shown as like small circles.
What is this image again?
It was an MR. It's D, right?
It's
electron microscopy. Yeah. Next, cytotoxic analysis showed that the IC50 of the virus was 104 MOI for NP53 cell line and 36 MOI for XFM cell line. Next, they again test the efficacy of this oncovirus in mouse with already established tumors. Where this nude mice? Forgot. There are so many mouse used. I totally forgot what kind of mice they use. These are immunocompetent models of mice that they are using right here.
This is the figure that makes this paper qualified to be discussed with in this podcast. I mean, in this journal club. Anyway, let's go.
Yeah, toxicity studies were done with viruses with dose ranging from 10 to the power 6 to 10 to the power 8 PFU for 15 days. And there was no lethality or toxicity observed via this virus in the cells, nor was any weight loss observed as compared to control mouse. So in figure A, they show that the tumor masses have high infiltration of CD3, CD4, CD8, and FOX, and a decrease in infiltration of FOXP3, which is also shown in graphs over here.
Can you explain what are those markers for?
Yeah, CD3 will basically be all the T cells which are infiltrating a tumor, whether they be helper or cytotoxic. CD4 is a marker for T helper cells only, and CD8 is a marker for cytotoxic T cells only. FOXP3 are marker for T regulatory cells, and T regulatory cells are anti-inflammatory cells. So we don't want anti-inflammation while we are treating something. So anti-inflammation will be like reversing the effect. Right.
Samriti, is that a question? Do you have a question?
Yeah, I just wanted to say that I really like this figure because a lot of papers, when they talk about CD4 cells, they forget about the regulatory T cells. And then we just sort of have to make an assumption, or well, the assumption that I would make usually is that, well, they're regulatory T cells, so that doesn't really tell me anything. But I really did like how they characterize the FOXP3 as well.
Yeah.
Yeah.
Yeah, that was interesting. So in these graphs, they showed significant differences shown by CD3. Actually, for CD3 as well as CD8, it's very much visible, the difference. Significant difference in case of CD4 too, and in case of CD8, and significant, not significant, but a little decrease in the T regulatory cells as compared to control. Next, they see the expression of mRNAs of interferon gamma, which should be specific to the virus. CD8A, which should be a subset of the cytotoxic T cells. CD4 in the treated tumor. And they do not, I think they take the 0 as the control or something, but they show that all of them have somewhat significant change in their mRNA.
So interferon gamma here is important to look at. I don't know if increase or decrease in CD8A and CD4 means anything. I'm not sure. I don't know if having a lot of CD4 helps these T cells in any way, but interferon gamma will be very useful because interferon gamma is great for activating natural killer cells. It's great for activating antigen presenting cells, so you can have the whole cycle of immunogenic cell death continue because of the interferon gamma.
Yeah, the mRNA doesn't seem to be a good representation of the cells which are right here, right, for CD8, because you can see so many number of cells which are infiltrating for treated mouse, but the mRNA is just a bit increased. So anyway, in figure G, the splenocytes were extracted from the mouse and co-cultured with tumor cells, and the splenocytes from virus-treated mouse showed higher expression of interferon gamma, which again, Jatin explained why is it important for a specific anti-tumor immune response. Finally, they are also showing some treatment in mouse models, which is necessary before you go for a clinical trial. You need to show treatment being effective in any animal model. In figure H, the tumor volume is seen to be decreasing significantly after the oncolytic virus therapy has been done. Then the survival analysis showed that the overall survival was increased for treated as compared to non-treated. To demonstrate the generation of immune memory, the long-term survival mouse were re-injected right here with more tumor cells. However, the mouse did not have developed any tumor after they have once been treated with the oncovirus already. So they say that yes, the immune memory is working after they are being treated with this virus.
Wait, 10% mice survived and those 10% mice did not die. That's what they're showing, right? That from figure I. That's
yes.
I don't know for with respect to cancer is I don't know how good of a survival that is. That looks like a very crappy survival.
It is at least better than no survival at all, which is shown by other like immunotherapies or other drug related therapies for this DIPG tumor for now. I think that's why they're so excited, because there was no treatment, but now there is some treatment. So this percentage of mice only, which were long-term survivors, were taken again in this study and then re-injected with the tumor cells, but there was no tumor formation even after injection. So even after above 40 days, there was no tumor, which is good.
I have a question. In the re-challenge experiment, so in the figure caption it says re-challenge experiment of the long-term survivor. So does that mean they only did this on one mouse?
Yeah, that's what I was saying. This 10% of whatever is surviving from figure I, that's what they're taking in figure J.
It says the long-term survivor.
I think I think they missed the S, but I won't assume that they did the experiment in only one mouse.
Wait, how many did they use in I? How many mice were there? So this is clearly 10% there. That's survival.
N is 10.
Okay, N is 10. So yeah, that means J is on one mouse.
Oops. Yeah, that's very, very suspicious.
Yeah.
Yep.
Well, it's somewhat working. Not my go-to treatment when I have glioma, but somewhat.
Strangely, it's been approved for clinical studies, so let's see what happens to human here.
Yeah. Well, they did the phase 1 trial I read, and it was safe. And they did convince somebody to give money for phase 2.
So this virus was already shown to be safe in multiple other tumor, but those were adult gliomas, and for the first time they are showing it in pediatric glioma. So I would have expected that they would be safe if they are safe in adult glioma already, but let's see if it is effective in the next clinical trial. Anyway, for this figure, they say that the treatment with virus does not help nude mice. So the nude mice die without giving any significant difference of change in survival. And it shows that one main part of the viral anti-tumor therapy is its following immune response, which is taking place. And that's why I was a bit shocked, because in the previous studies which were done on nude mice, the virus itself was giving like 33% of survival. And suddenly here they show that there is no significant survival at all,
But the
which was a
previous
bit
studies were
confusing.
from human cancer cells in mice.
Yes,
So this is
yes. Mouse
mice.
cancer.
Yeah, yeah,
Yeah.
yeah. Well, it also
But
maybe
uh,
this. Yeah, I don't know exactly, but
does it
maybe
make
it
any
it would
difference?
it would work better on humans than mice. Another thing I want to mention here is that, well, that one mouse did better. So that one mouse who survived in figure I wouldn't be surviving in figure K. So that one mouse is has a good immunity thanks to this virus. But I also want to point out that a decade or few decades ago, we thought that the blood-brain barrier doesn't allow immune cells to go to the brain, and now it looks like a lot of tumor immunity inside the brain depends on the immune system. So it's pretty fascinating that
It
they go
is.
there, and I must have a very tight regulation, but they are important.
Mm-hmm. A few years ago, no one would expect using virus as a therapy too for cancers.
Yep.
Right? This is a upcoming field, and I'm so fascinated by it. Okay. So Delta-24-RGD can elicit antitumoral immune response in vivo, which plays a crucial role in clearing up the tumor. So yeah, their animal model as well as in vitro studies were successful, and that's why they got permission to do carry on clinical trial phase 1 and phase 2 for now.
Successful,
Yes,
the
let's see.
term.
Um, okay. So in discussions, they mentioned that RB1 mutation is more frequent in hemisphere grade, high grade gliomas, which was pHGG, than brain stem tumor, which was DIPG. In general, too, as I told previously, DIPG is not curative till now, and DIPG is much more difficult to treat or detect than pHGG. And it was interesting to show that the Delta 24-RGD was increasing the infiltration of lymphocyte to the brain and opening this opens up multiple combinatory strategies with immunotherapy. And this is pivotal in this finding. This is a pivotal finding in this area of pediatric tumor, because immunotherapies by themselves have showed limited results due to incapacity of crossing the blood-brain barrier and reaching the tumor site, which is already having a immunosuppressive microenvironment surrounded. So yeah, microenvironment in it, but due to this virus, somehow the blood-brain barrier is allowing infiltration of immune cells. And maybe if you give any other cytokines or stimulatory factors, maybe the infiltration will be much higher. This paper concludes by saying that Delta-24-RGD infects, replicates, and kills DIPG murine uh, tumor cells. I forgot mentioning the tumor right here in vitro. And the in vivo administration of Delta-24-RGD triggers an immune response against the DIPG tumors, leading to survival in mouse model. And I would open up for any other questions. Otherwise, I'm done.
That was a very different paper for me. Thank you for choosing this. I get to read about things that are outside my comfort zone. Tanu, why did you choose this, by the way?
So I'm working with norovirus, but I'm not working much on the immuno side of it. I'm working how the interaction of norovirus and microbiome helps the host overall. So there is some immunology, but not much immunology. But I'm very, very interested in this kind of studies, even in the studies where bacteriophages are um, used against the pathogenic bacteria in our gut or in our oral cavity, and in these studies where the oncolytic viruses are used against tumors. So I really like how people are taking advantages of viruses which were thought to be a very negative thing, and people are now using it for helping humans, help for medical field.
I thought adenoviruses were they were used for gene therapy, right? Because they can integrate inside in the genome. There is no integration happening in this case, right? This is just straightforward killing.
Yeah, this is only the lytic phase, not the lysogenic phase, so no integration is happening in this
And
adenovirus.
do you know why specifically? So because I am bad at with viruses, I don't know much. Why adenoviruses? Why not any other? Why not a retrovirus?
So also this is a replication competent adenovirus where I would guess they have removed some genes for integration, but I'm not very sure. And I don't think retrovirus will be effective, and
Don't they kill? They also kill, right?
the retroviruses mostly undergo the lysogenic phase.
No, they have to kill. Else, how does HIV kill the T cells?
So even HIV is a latent infection. It stays in your cells for multiple years, and then when it gets the perfect conditions or when it wants, it will form progenies which will start killing you. It's not that as soon as you have that virus inside you, it just starts killing.
And can RNA viruses be used for the same purpose? Just from your background, do you think so? I know none of us are experts at oncolytic viruses, except Samriti, she is.
Yeah.
You better be, you
For
have to graduate soon. No,
so
I'm
for
just kidding.
our
Nobody is an expert. I sometimes think even professors don't know as much as people expect them to.
I think that's what you learn in grad school, right? Then you don't know anything.
Yeah.
So Jatin, one problem with the RNA virus is its rate of mutation. So I really doubt if you do any lab mutation, like lab induced mutation in those viruses, they will actually go in the host and stay like that. They can turn into infectious viruses very, very soon. And that's one negative point of oncotherapy. You don't want the virus to turn infectious.
Is that because of RNA polymerase, the RDRP, that the RNA dependent RNA polymerase these viruses have? Is that not good at
That's
proofreading?
faulty. That's not good at proofreading.
Okay. Okay, I like this paper. Anybody else who has questions?
Yeah, I have two comments.
Mm-hmm.
Both of them have to do with the translation of this therapy to humans. The first one being that all of the studies that they did in vivo, they did intratumoral injections, right?
Yes. Yeah.
So doing that in humans would be very difficult if you're looking at something that's, a glioma, like a brain cancer.
Mm-hmm.
So intratumoral injections, I don't think. I mean, I'm not sure. I don't work with brain cancers, but what I would think is that would not really be possible. So with any oncolytic virus that you're targeting to the brain, I would suspect that it would mostly be intravenous injections, right? So that poses a lot of different obstacles in the way, the blood-brain barrier just being one of them. But for the adenovirus to reach the brain, I could foresee there being a lot of problems if you are doing it through the intravenous route. And then the second problem I see is that adenovirus is a very common virus that's just in our environment. And so what that means is that a lot of people can be seropositive for adenovirus, meaning that they have pre-existing immunity. They already have antibodies and things like that to adenovirus. Even I think just in the North American population, there's about 30 to 40% of just the normal population that it has pre-existing immunity to adenovirus. And so that also will limit, I would say, the effect of this therapy in patients as well, because the virus probably won't even be able to do what it needs to do, because it will probably be cleared by the immune system before it's able to function.
So for your second comment, I think if they are administering the virus intravenously, then maybe there will be an immune response. But if they are, I don't know how they will, but if they are directly giving it through the spinal cord or above the blood-brain barrier, I don't think there will be an immune response. But the question is, how will they give it directly in the brain?
Yeah,
I'm not clear. Yeah.
Maybe they'll need to change some epitopes on this virus.
I don't know if they do something like do a surgery where you remove your skull, inject it intratumorally, and then I don't know if they are doing something like that.
I don't know. But to Jathan's point about changing the epitopes, if you're doing that much to make this one virus work, at that point, wouldn't you just choose a different virus?
Yeah, that also makes sense.
Yeah.
Isn't that why people use adeno-associated virus, the AAVs, instead of adenoviruses, because of the seropositivity?
Mm-hmm.
Maybe they'll change it.
Is the adeno-associated virus for oncolysis, or is it just for gene therapy? I thought it was just for gene therapy. It's a better choice.
I'm not sure.
It's being studied now for its oncolytic properties.
Okay. I know it's good for gene therapy because they don't integrate any your genome, but they form a episome in the nucleus, which is a genome of their own. So they are not basically disrupting any functional genes that you have.
Any other questions? Okay.
Nope.
Tanu, I really liked your presentation. Which template did you use? I mean, I like the graphics. You put in a lot of effort on this.
This was suggested by PowerPoint itself. I'm stopping the sharing thing.
Okay.
Yeah.
And next, Anthea said she will be presenting the next paper, right?
We can discuss it in GroupMe.
Yeah, we can discuss that in GroupMe, and we can also decide the next date when we meet. And for our podcast, we are discussing gene rearrangement. Anybody who is good at gene rearrangement and want to join as a guest, you are free to join me, because I'm going to have to read a lot of that gene rearrangement in B and T cells. But yeah, thank you all for joining, guys. And thanks anybody who was watching.
Thank you, everyone.
Okay, I'm going to unmute all of us. Bye-bye.